Asmaa Huessin, and today it would be such a pleasure to deliver a presentation simply on genome editing. Let me ask you a question first. What do you think genome editing, or gene editing as it is also known, is, and how does it work? Well, let's start talking about genes. These are sections of the long DNA molecules coiled up inside each cell of every living thing, from microorganisms and insects to plants and animals, including humans. Think of DNA as the instruction manual for an organism. In people, genes can influence characteristics such as your height, the color of your eye. And as we all know, DNA is inherited from our parents. We have a combination of two sets of genes, one from each parent, brought together to produce a set of instructions, reshuffled in each generation. But hold on for a sec. Sometimes these instructions may carry errors. These can cause problems. Ladies and gentlemen, faulty genes cause serious illness. In a matter of fact, humanity has a long history of figuring out ways to modify genes. They have been crossbreeding plants to make them better to eat for thousands of years. But with genome editing, we have made a great leap forward. This new technology is faster to use, relatively simple and cheap, comparing with the previous methods. In a nutshell, it works by identifying, then cutting pieces of DNA. One way of doing this uses a component known as CRISPR to pinpoint the exact DNA sequence to be altered within the gene. Then another enzyme called Cas9 snips through the DNA, changing it or allowing it to be replaced by another DNA sequence that is introduced at the same time. It's like a find and replace for the genetic instruction manual. Actually, by making these microscopic changes to DNA, gene editing can make big changes to our lives. Gene editing is already transforming genetic research, but at this early stage of such a powerful technology, it is crucial to weigh the possible benefits against the possible negatives. Let's start by talking about the benefits. We can say that gene editing can make radical improvements to human health. For example, in 2015, doctors used genome editing techniques to cure a baby girl's leukemia. And also in the future, we may be able to fix mutations that predispose to cancer and enable new therapies to HIV or edit genes that cause hereditary diseases. And as for the plant world, crops can be more nutritious, disease-resistant, and able to grow in difficult conditions. Actually, scientists are working currently on the genes of mosquito to prevent them from carrying malaria. Okay, but what about the possible downsides? Well, see, genes could be edited in early human embryos to alter some characteristics that have no bearing on health, like the eye color, which affects descendants too. And also keep in mind that these techniques will also develop more violent microbial diseases. So now it's obvious to all of us that alongside the benefits, there are some ethical and societal concerns to consider. The technology is already in our hands. Now it's time to debate how to use it, regulate it, avoid the negatives, and unlock the potential positives to provide life-changing solutions to the most or the world's biggest challenges. Thank you.